Okay, so in the last half hour to 40 minutes of this lab activity, there's a necessary bit of discussion that needs to happen. The view from 20,000 feet, where you can put everything you've done today into perspective. Usually, by this time, people are done with their actual simulations, and a few groups are actually done with all of their plots. A few people are still working on them, but I would walk around class and grab some of the graphs from people that are done and project them on the big screen for everyone to see. First, there's simulation A, which by all accounts should give us a classic type 2 survivorship curve, a straight linear decline. The likelihood of survival or death doesn't change over the entire simulation. Now, does everybody always get a perfectly straight line? No. That's what happens when you're looking at a real system subject to the vagaries of chance sampling. The departures from linearity are almost always going to be more pronounced on the right-hand side of the graph, where the finite sample effects are the greatest. One group might have a sharp downturn because out of the 4 cubes they had left, 3 of them died in a single roll, while another group might see their curve flattening out because with a small number of cubes, it's possible for them all to survive for several turns in a row. And we actually saw that in the recording of my rolls for simulation B. This is why it's actually okay to disregard data based on a small number of survivors, because random variations are going to have a disproportionate effect on them. But I would say that in this activity, everyone either gets a type 2 plot or understands why it should be a type 2 plot. Not getting the precise expectation here is fine if you understand why it should have been a linear decline and why deviations from the expectation are also expectable. In simulation B, the graph should look like a classic type 3 age-specific survival curve, starting at the natural log of 500. Your plot, based on the data you watched me generate, might have fewer points than what most students would be getting in class, because as I remember, there was a rapid decline after year 2, and we reached the stopping point of 2 individuals in only the next couple of years. But even so, there is still enough data to give an expectable type 3 concave shape that you would see with animals with very high juvenile mortality. So in simulation C, the first challenge you faced was just getting your data into a format that was actually usable for plotting age-specific survival. Did you start by creating evenly spaced age benchmarks, the equivalent to years in our earlier plots? For example, instead of 0 years, 1 year, 2 years, 3 years, you could have used 0 seconds, 15 seconds, 30 seconds, 45 seconds, 60 seconds, 75 seconds, 90 seconds, etc. Depending on your actual results, you might have needed to adjust the size of your intervals based on the range of your data. So if your bubbles lasted between 0 seconds and 3 minutes, you might choose a larger interval, say every 30 seconds. Well, you could go with a shorter interval if your bubbles were shorter lived. If they mostly died before 40 seconds, you might want to census your bubble cohort every 10 seconds or so. I'd be willing to bet that your indoor bubbles ranged into the longer ages more so than the outdoor bubbles. Also, in your graph, you should probably stop plotting when your number of survivors gets down to 2 or 3. Any lower would not be informative. At 1 survivor, the natural log would be 0. And at 0 survivors, the natural log is negative infinity. And so there's absolutely no point in including that last survivor. Moreover, by now you should also know what happens when you have a tiny number of survivors. You're not going to be getting a representative picture of the survival probabilities anyways. Now, what we typically end up with is a plot looking generally of this shape, with the indoor bubbles showing a type 1 pattern with a generally longer life, as compared with the outdoor bubbles, which usually follow something closer to a type 2 shape and also dying out much quicker relative to the indoor bubbles. Now, having done the exercise yourself, you can probably easily explain why the outdoor bubbles that were freed to ride on the coronavirus infected wind would live for a shorter time. And maybe you can even rationalize why the shape of the age-specific survival plot is somewhat linear. Is constant survival, constant mortality a reasonable proposition for these bubbles? Why might the survival rate for outdoor bubbles be unaffected by the age of the bubble? A somewhat harder question to address is why would the indoor bubbles show a type 1 pattern? Clearly, lacking the freedom of their outdoor brethren, they have a more sheltered existence and are not subject to the many causes of death encountered by the wild bubbles. Yes, this is actually very similar to a comparison between a captive population of zebras and a dazzle of zebras in the wild. Wild zebras may be subject to a relatively steady and high rate of mortality from various causes that are just absent for a captive population in a zoo, like the San Diego Zoo Safari Park, where we would be going in 2 weeks if not for the coronavirus conditions. Now, in a zoo, the animals are well attended to. None starve to death. None are taken out by lions. If they get an infection that would be lethal in the wild, instead, they get proper veterinary treatment and are much more likely to survive. Both captive zebras and the captive bubbles that we recorded data on indoors are allowed to die of natural causes much more frequently than ever occurs in the wild. That part is relatively easy to understand. Of course, the captive bubbles live longer lives. The harder question we need to address now is why the curving downwards. Why is death by natural causes something that becomes progressively more likely with advancing age? In the case of soap bubbles sitting on a wand, death happens when the continuous film of soap and water surrounding the air mass loses integrity and becomes discontinuous. This happens when the film, usually on the top half of the bubble, becomes extremely thin. And this would be due to two factors, one being gravity, which would draw the soapy solution downward. And this is why the bottom half of the bubble is not the part that initiates the popping. The second factor is evaporation. The soap film of the bubbles you blow all start out at about the same thickness, and the rate of evaporation is relatively consistent for the still air inside your room. Drying out to a critical thinness is going to happen within a predictable time range. By the time we get to the median time to critical dehydration, the rate of bubble death should be peaking. In other words, mortality starts out low and gets higher, resulting in the type 1 curve. That makes sense enough for bubbles, but the same phenomenon occurs with captive zebras. Or, closer to home, we can point out that humans living in the modern world are very much like captive zebras, largely protected from the major causes of death that probably afflicted our ancestors from not that many generations ago. Go back a few thousand years, and it's entirely plausible that the age-specific survival curve for humans would be type 2-ish, like your wild bubbles. People would mostly be getting killed by something by age 35, and the few living past this age would be a rare oddity that couldn't be terribly consequential for shaping human evolution. Today, we see the higher rates of mortality in the 60, 70, 80 year and beyond ranges only because we actually have a lot of people living to those ages. The strange twist that I'll introduce here is the idea that declining health late in life is something conferred upon us by our genetics, and that the evolution of these old age problems may actually have occurred during the millennia before anybody ever lived to those ages. Let's start with the premise that declining health late in life is genetically conferred. What evidence is there for this? Well, in fruit flies, this is research that I was involved in as a postdoc, it's actually relatively easy to impose artificial selection on flies to make a strain in which individuals live and are healthy well past the 80 days, which is the norm for fruit flies. Okay. Now, what about in humans? Do you know of any families in which long, healthy existences well into the 90s and sometimes beyond is actually the norm for that family. Now, if you do not belong to such a family, and thus your expectation of lifespan, health span is not so great, then that suggests that your family carries a greater burden of genes causing poor health late in life. Genetic variability for longevity exists in flies, and I would argue that based on our own experiences, similar variation exists in humans as well. And by saying that humans show a type 1 age-specific survival pattern, basically acknowledges that senescence, the expectable deterioration of health with age, in humans is a species-wide characteristic. And I don't mean to say that it happens with every human, but rather it's a pattern that characterizes the human gene pool. And this indicates that the genes causing declining health at advanced age are widespread and prevalent in the human species. And this seems to present a paradox. We know that natural selection favors higher survival, not lower. And these genes are clearly resulting in lower rates of survival, albeit late in life. Now, at this point in our discussion, someone unfailingly argues that such genes have evolved for the good of the species, because by killing off the old, this frees more resources for the rest of the population, who then enjoy higher survival and reproduction. Does that sound like a reasonable argument to you? It's not. And let me be very clear in my explanation why. The beneficiary of a selected gene must be the holder of that gene. If I have a gene that causes me to be particularly successful at surviving, I am the one that benefits, not the rest of my species. I'll survive better than most, have more offspring than most, and that gene will be at a greater representation in the next generation as a result. If I'm the first one in my species to have this gene, natural selection will be a real force that increases the frequency of the gene until it becomes prevalent, if not fixed, in the human species. Now, if I have a gene that guarantees me a fatal heart attack on my 60th birthday, it might actually be true that the species does better with my self-snuffing. And it might also be true that the human species would be better off if everybody had this gene. But if I'm the first one in my species to have this gene, how could natural selection cause it to increase in frequency unless I actually had higher fitness as a result of having this gene? What would be the benefit to me from having such a gene in my genome? Now, I'll say this in all caps. Nothing ever evolves for the good of the species. This is an unfortunate canard, and if you ever use an argument based on good of the species, you're basically advertising in big, bold letters that you don't understand how natural selection works. Next time you hear someone say, it evolved for the good of the species, I want the hairs on the back of your neck to bristle as you get ready to slap whoever said it. So we can put this argument to rest, 6 feet under where it belongs, right? Good. So now, if not that, how could we account for the abundance and diversity of genes in the human genome that have negative effects on health that are expressed at advanced ages? Now, I want to offer two explanations. The first takes into account our long history before the advent of modern medicine. Any gene whose effect doesn't occur until age 60, good for health or bad for health, would be completely inconsequential in a human population in which no one ever lives past the age of 40. Such a gene could arise and increase by random genetic drift or even become fixed without anyone ever being affected. Until, that is, people start living to that age, for example, in a later era. Now, the second explanation is a bit more involved, and it introduces the concept of life history trade-offs, which is a topic that we address in lecture in the last unit of BIO202. Is it possible for fitness at two different times of life to be affected by the same gene? Specifically, is it possible for health late in life to be negatively correlated with, say, reproduction early in life? Now, I chose this example because it's directly pertinent to our current question of how humans could have come to have high frequencies of genes causing poor health late in life. Well, if those same genes caused higher fitness early, then you could say that positive selection on the fitness benefits early in life are overriding the negative selection occurring against those genes because of the health problems they cause late in life. I would argue that we have good reason to think that early reproduction and lifespan may actually be negatively correlated. You can think about how this would work even without bringing genes in at all. Compare identical twin females. Twin A has two children in her 20s, while twin B starts making babies at age 14 and has her 18th child at age 42. Now, gestation and childbirth are physiologically really taxing things. All else being equal, would you expect these two sisters to have exactly the same lifespan? In some ways, having offspring is just like cigarette smoking in the way they shorten lifespans. Each baby might be the equivalent of a couple of cartons of cigs, X number of years cut from the end of your life. Now, you may not like this analogy, but I hope that you can see its substance. In the case of our identical twins, the genetics are obviously not the issue. Presumably, it's a personal choice of individuals to either restrain reproduction or to go nuts making babies. And so the differences in reproduction and longevity are not heritable, not caused by genes. But now, what if you had a gene that caused a greater drive to reproduce early? Women with this gene average 1.3 children more than women lacking the gene. Wouldn't you expect this gene also to be associated with a somewhat shorter lifespan? This is an example of a life history trade-off. It would be nice to have the ability to maximize both lifespan and reproduction. That would be ideal. But the reality of organisms says that there's going to be a limited availability of resources. And at some level, one must choose between allocating either towards reproduction or towards self-maintenance and survival. You can't do both. More resources allocated towards reproduction means that there's less left for survival. And a more conservative allocation that ensures survival means that you don't have as much to put into reproduction. This is a central concept behind the trade-off, and it's what sets up ecology to be a major force driving the evolution of life history traits of organisms. And that's what we'll be developing in lecture very soon.